Hello, and welcome to Lesson 5.5. My name is Chris Durland, and I'll be talking you through the wrong answer types that you are going to see in the science sections of the MCAT. Our learning objectives will be twofold. The first revolves around the idea that very often answered choices have a repetitive structure. We'll see that we can actually use that to our advantage. The second learning goal revolves around the idea that while astute, the AAMC and the test writers they employ are not always the most creative when it comes to crafting wrong answers. So in the second half, we'll take a look at the wrong answer types as they manifest in the science sections. Our primary focus will be MCAT expertise, that fifth element that the MCAT tests. We'll be looking to hone this skill within the science concepts of oxidative phosphorylation, factors affecting genetic variability, and the endocrine system. At this point in your studies, you've no doubt come across a question with answer choices that vary with as little as one word. This presents some questions. Why do the test writers include repetitive answer choices? Give you a clue, it's not always because they're lazy. What should we focus on when we see repetitive answer choices, and how can we use that to our advantage? We'll start off by looking at some questions with answer choices that exhibit minimal repetition, and then we'll contrast that with some typical repetitive answer choices. Take a look at number 1, which at first glance exhibits very minimal repetition. However, as we examine the answer choices a little more closely, we do see there is an element of repetition. They all share the suffix "ase," indicating that we are dealing with enzymes. Further, as we begin to classify these enzymes, we'll notice that they are all involved in respiration at one point or another. The key to answering this discrete question is, of course, which enzyme. We are asked about the last enzyme in the electron transport chain. So thinking about the electron transport chain, you may have the last enzyme memorized, in which case you've got one more correct answer on test day. However, if you don't have this memorized, you can still reason through it by thinking about the processes involved in the electron transport chain. So the electron transport chain is basically a series of redox reactions where high energy electrons are passed to molecules with increasing reduction potentials. And well, what is waiting there at the end? Oxygen, coming from the very air we breathe. So oxygen is the final electron acceptor, and we're looking for an answer choice that's going to reflect that, and we see an oxioreductase in D. This is a great example of how a little bit of outside knowledge and a lot of reasoning will go a long way on the MCAT. Taking a quick peek at the answer choices for number 2, what do you notice? There's not a whole lot of repetition in here, granted. We're dealing with fractions, and there may be a pattern within the fractions, and we may be able to use that to our advantage, but in terms of repetition, not a whole lot. As we begin to think about our plan, it may be tempting to try and quickly do this one in your head. However, it's not necessary. We have scratch paper, and this one's probably going to be better off attacked by drawing out the different stages, mutating a chromosome, and following that mutation through the different phases. And so by test day, we ought to be able to come up with a diagram much like we have on the right-hand side of our page. So familiarizing ourselves with this diagram, we can see that we have a simplified karyotype. If we were dealing with the human karyotype with 46 chromosomes, we would need pairs for the letters A through W. But as you can imagine, that would be a little time-consuming on test day. We can also see that we're dealing with a diploid spermatogonium. We have an uppercase A and a lowercase a, meaning that they are homologous. They code for the same genes, one from mom, one from dad. So let's go ahead and mutate one of our chromosomes. We'll just mutate the chromosome indicated by the capital A. It is the spermatogonium that will undergo DNA replication, in which case we are going to copy all of our chromosomes. Well, what does that mean for our primary spermatocyte? Well, if we had a mutated chromosome in our spermatogonium, it would be replicated and would be found in 2 sister chromatids of a primary spermatocyte. In meiosis 1, we get segregation of homologous chromosomes, meaning we will separate the uppercase letters from the lowercase letters. So both of our mutated chromatids will end up in the same secondary spermatocyte. In meiosis 2, we separate the chromatids from each other. So this mutation is going to show up in 2 of our 4 sperm cells, or half. So as we can see, there really wasn't a whole lot of repetition in these answer choices. However, there is some repetition in the content. This question was asking about spermatogenesis, but on test day, you could have very easily been asked about oogenesis. And the thing to keep in mind is that they share the same naming template. We'll start with a spermatogonium or an oogonium and go to a primary spermatocyte or an oocyte, and then a secondary spermato-ooa, and then a sperm or an ovum. So keep in mind, repetition is your friend. Now that we've seen some questions and answers with little repetition, let's compare it to a typical repetitive set of answer choices. What do you notice about the 4 answer choices? We essentially have 2 classes of receptors, and we have 2 possible combinations, either embedded in the membrane or dissolved in the cytosol. The specifics of these receptors are outside the scope of required knowledge for the MCAT, meaning we're going to need some clues from the question stem. As we read through the question stem, we'll notice that it starts off in a typical AAMC fashion where they throw some extraneous knowledge at you just to intimidate you. Don't worry, the MCAT tests the basic sciences. We'll need to keep reading in order to find the relevant clues. As we continue our search for clues, we come across this fact. The GPR30 receptors are G protein coupled receptors. Now, that is something that we should walk into test day knowing. These are membrane-bound receptors. How does that help? We can eliminate any answer choices that indicate otherwise. So there goes A, and there goes C. All right, I feel better about this question already. At this point, when we compare the remaining answer choices, we can see that these receptors are either intracellular, as indicated in B, or on the cell membrane, as indicated in D. In order to determine the location of our receptor, we're going to need to know a little bit more about the hormone in question. You may recognize estradiol as a steroid hormone, but if you didn't, they gave us a clue in the very first sentence where they say that testosterone is converted to estradiol. We should know that testosterone is a steroid hormone, and that if it is converted to estradiol, that estradiol is most likely a steroid hormone as well. How does that help? Well, steroid hormones are lipid soluble, meaning they diffuse fairly readily from the blood through membranes and into target cells. So if our hormone is permeable, we're going to expect our receptor to be intracellular, and there's only one answer choice that indicates that, B. Notice that just because we had repetitive answer choices, it doesn't necessarily mean this is going to be an easy question. However, as we begin to pay attention to the repetitive structure, it allows us to dissect these seemingly intimidating questions and reveal them for what they are. In this case, we had 2 rather straightforward questions. Where are G protein coupled receptors found, and where are receptors for steroid hormones? So the key to using the repetitive structure is to use what you know, a fact, a clue from the passage or question stem, to eliminate answer choices that are inconsistent and to begin to really focus in on the relevant factors and questions being asked. Take a look at number 4. Wow, lots of repetition in these answer choices. Upon closer inspection, we can see that we're dealing with the 4 possible permutations of increase and decrease for the mitochondrial matrix and intermembrane space. Number 4 asks about the pH change during the generation of the proton motive force. Recall that during the electron transport chain, those high energy electrons have extra energy free to do work. In this case, we harness that potential to pump protons across the membrane. So at this point, since we know that protons are pumped from one side to another, we know that the pH of one is going to increase and the pH of the other is going to decrease. Using that relationship, we can eliminate a couple answer choices that say that they both increase or decrease. And this is where knowledge of the anatomy of a mitochondria comes into play. The protons are in fact pumped from the matrix into the intermembrane space, thereby creating an electrochemical gradient, which is then harnessed by ATP synthase to phosphorylate ADP into ATP. This process of using a proton gradient across a permeable membrane is known as chemiosmosis, and it's a fundamental part of oxidative phosphorylation. So the protons are pumped out of the matrix, meaning the pH is going to increase. Necessarily, the pH of the intermembrane space will decrease, and D is our answer. Notice in this one how we used a pattern within the answer choices to eliminate a few. The savvy test taker knows that the right answer must not only be in agreement with the question stem, but the different parts of the right answer must be in agreement with each other. Number 5 presents us with another set of answer choices with a repetitive structure. Notice in this case, we're talking about homologous chromosomes or non-homologous chromosomes and mitosis or meiosis. You may also want to note that prophase 1 is in all of our answer choices, meaning it's not a relevant fact. You may also be thinking that only one form of reproduction necessitates the Roman numeral 1 after the prophase, and if you are, feel free to use that to eliminate some answer choices. The question stem refers to crossing over, and that is indeed a buzzword for one of these types of replication. However, it also says that it is a contributor to genetic variation, meaning it must occur in the germline. Another big clue pointing in the direction of meiosis. We can use that to eliminate the answer choices with mitosis. We're still left with the question of homologous chromosomes or non-homologous chromosomes, and it should make sense that we have crossing over between homologous chromosomes, those chromosomes that code for the same genes. This way, there are no genes that are lost or duplicated. Crossing over between homologous chromosomes is significantly less likely to lead to missing or duplicate genes. So as we can see, sometimes test writers craft really similar answer choices to add another layer of difficulty. Whenever you see two or more answer choices that are nearly identical, it's going to be in your best interest to determine exactly how they're different. Very often the right answer hinges on those subtle differences. Every single question on the MCAT will have 1 right and 3 wrong answers. Where do these wrong answers come from? How do test makers generate them? Those will be some questions we'll be looking to answer in our second learning goal today. To facilitate our discussion of the wrong answer types, we have a fairly open-ended question. Which of the following occurs in a normal, healthy adult male? It's general questions like this that make predictions difficult. Generally, process of elimination will prevail. And that's what we'll do with this exercise. In fact, all of these answer choices are wrong. Our goal will be to determine which pathology or pathologies each of these answer choices exhibits. Take a look at the first one. The content looks pretty good. What's wrong with it? It's really just a single word, a qualifier, the word only. Is the portal system in question, the hypophyseal portal system, the body's only portal system? No. In fact, we should be familiar with the hepatic and the renal portal systems as well. So this is not the only portal system, and this answer choice is too extreme. Notice the degree to which the content is tested here. Not only do we need to know the details of ADH stimulus, synthesis, and transport, but also about the different portal systems. If you're not sure, this is a pretty tempting answer choice. While extreme answer choices are not always wrong, they should raise a red flag. Oftentimes, the extreme statement is going to be the easiest to disprove, which is why it's usually a good place to start. For instance, in this question, we don't even need to consider ADH, really just the portal systems of the human body. Take a look at our second answer to see if you can determine what's wrong with it. Once again, the content seems relevant, but here the order is mixed up. The Sertoli cells are stimulated by FSH, whereas these cells of Leydig are stimulated by LH. So this would be an example of an opposite. You'll see lots of opposite answer choices, and they're tempting because the content is so relevant. Oftentimes, it's a single word that will throw these off as well. Then we have our third wrong answer. See anything wrong with it? Well, there's nothing factually incorrect about this statement. It's a true detail about male sexual differentiation. So why is it in our wrong answer exercise? Perhaps a reread of the question stem is in order. Notice this true statement does not adequately answer the question. Development of the Wolffian duct does not occur in an adult male. We might classify this wrong answer as a faulty use of detail. These will naturally be more common in passage-based questions, but the temptation is the same. You're presented with a recognizable fact, but just because you read it in a passage or in the review notes doesn't mean it correctly answers the question at hand. Reading the next answer, we see a similar pathology, so this one would have elements of faulty use of detail, but just taking a step back, we're talking about a normal, healthy male. Pregnancy should not even be a consideration. It's simply out of the scope of this question. Our next distractor, which is actually the AAMC's word of choice for a wrong answer, deals with negative feedback. We should probably wake up on test day knowing that iodine is necessary for thyroid hormone synthesis, which is, of course, why it's in our salt. So without adequate iodine, what happens? In a normal, healthy adult male, that is. Since we're dealing with negative feedback, it is the presence of the thyroid hormones that will feed back in a negative manner, thus inhibiting the pathway. So what if we can't synthesize a necessary hormone? Thyroid hormone levels will drop. Thyroid stimulating hormone kicks in as an attempt to raise thyroid hormones. So this wrong answer classically mixes up an increase versus a decrease, which is why it's an opposite. Our final wrong answer says, after a meal, insulin is secreted by beta cells in the pancreas, thereby maintaining elevated plasma glucose levels. So we're dealing with more endocrine regulation here. Glucose levels rise after a meal. This, of course, stimulates a hormonal response to bring blood sugar levels back to normal. The hormone is, in fact, insulin, and it is secreted by beta cells. But insulin lowers plasma glucose levels, so this one is opposite. However, you may have noticed the true detail in the first half of this question stem and thought, faulty use of detail, and you wouldn't be wrong. Furthermore, this answer implies some sort of pre-existing condition of elevated plasma glucose, which is, of course, out of the scope of this question. We don't know anything about the plasma glucose levels before the meal, and if they were elevated, that might be indicative of an abnormality. So what's the takeaway? Some answers are just train wrecks with pathologies that spill over into multiple categories. Don't let this distract you. On test day, you only need to identify a single flaw with an answer choice before eliminating it, by striking it through, of course. These wrong answer types are nothing new. You've already seen them covered in the analogous CARS lesson, which is good. You don't want to have to memorize a whole new set of terminology or switch languages when you're navigating from one section to the next. We can use the same Kaplan terminology in all 4 sections. The last wrong answer type is unique to the science sections. Take a second to assess this question and perhaps begin to think about what a plan should include. The answers indicate we'll probably need to do some math. The stem provides half reactions and relevant reduction potentials, as well as an equation to plug the reduction potentials into. At this point, you may be thinking triage, and that may not be a bad idea on test day. However, today is not test day, hopefully. So let's walk through this one with one key thing in mind. Numerical wrong answers are not randomly generated. They are thoughtfully crafted with common mistakes in mind. Let me show you what I mean. We're going to walk through this entire problem together, noting common mistakes that lead to miscalculation wrong answers. The question asks about the energy released by the system, which will be the sum of the energy released by all of the reactants. So we need to know the reaction taking place. We know that NADH is oxidized, or in other words, it's losing electrons. If NADH is oxidized, then oxygen must be reduced. Now the reduction of oxygen is shown in the top half reaction. However, the bottom reaction shows the reduction of NAD+. To obtain the oxidation of NADH, the direction of the reaction will need to be reversed. Putting the two together gives the reaction we see here. To calculate the change in potential for our system, we need to combine the reduction potentials for each half reaction. This is a common place for mistakes. The reduction potential for the oxidation reaction should be subtracted from the reduction potential for the reduction reaction. It's easy to get that confused, and unfortunately, if you do, and then do everything else correctly, you'll end up with the wrong answer, A. Plugging in the correct values, we obtain the potential change for our system, but that doesn't mean we're in the clear. Oh no. When plugging in values for the Gibbs free energy change, we need to recognize that 2 moles of electrons are being transferred for every single mole of NADH oxidized. If I were to plug in 1 and then do everything else correctly, I'd unfortunately end up with B. Using the correct value, we obtain the energy released per mole of NADH. Okay, so now we have the energy released per mole of NADH, and we just need to divide by Avogadro's number to get the energy released per molecule. Finally, we have the energy released per molecule. However, if you're not mindful of units and go looking for a match, again, you'll find a wrong answer that matches. Only after converting from kilojoules to joules will we have arrived at the correct answer. So I'll be honest, this question is on the more demanding side of the spectrum, but it's not unfair. In fact, each mistake was a simple error, one that we should be identifying now while we're practicing, so that when the real deal rolls around, you'll know what to be ever so mindful of. One more point to make, which addresses the Kaplan tip at the bottom of the page. When you look at the answer choices, what do you see? When I look at C and D, answer choices that differ by 3 orders of magnitude. Naturally, any answers that differ by orders of magnitude should alert you to common mistakes made while manipulating scientific notation, but 3 orders of magnitude is representative of another common mistake, one that should get us thinking about units. Furthermore, when comparing B and C, I notice they differ by a factor of 2. Recognizing this allows me to begin to predict where this may come into play. You can bet when determining the number of moles, I'll give it a little extra time. Because who wants to go through all this trouble only to come up with a wrong answer that you don't even know is wrong because you found a match? It's not until 30 days later when you get your scores back did you figure out that something went wrong. And actually, when you do get your score report back, it doesn't provide itemized feedback, so you may never know what went wrong. The takeaway here, learn from your mistakes now and anticipate the same traps in future problems. Okay, Now that we're warmed up and thinking like test makers, let's take a few minutes to outline our next passage, which after a quick scan appears to be about chromosomal mutations. This one's all you, so take a few minutes to build that outline and then we'll discuss. The first paragraph provides us with a bit of an introduction. We're going to talk about chromosomal abnormalities, and they're going to be classified as either structural or numerical. Your passage outline should include something along those lines. When creating your passage outline, don't just mindlessly jot down a few terms. Instead, use it as an opportunity to step back, anticipate what's going to come. For instance, our introductory paragraph talked about 2 types of chromosomal abnormalities. What do we anticipate the following paragraphs are going to be about? You guessed it. Paragraph 2 is all about structural. Paragraph 3 and 4 were about numerical. Your passage outline may have also noted the mention of translocations, their classification as either balanced or unbalanced, as well as a reference to Figure 1. It's always important to pay attention to figure references within the text. Very often when we struggle trying to interpret a figure or a table or a graph, it's because we missed something from the passage itself. The third paragraph is, of course, about numerical abnormalities. The paragraph also discussed partial trisomy as well as some potential causes. The final paragraph was all about trisomy and monosomy, and it provided a lot of details. It's not important to memorize these details, only to acknowledge and label their location. When presented with a figure, the experienced test taker will look for a title, read the caption, and always try and make connections to the passage. In this case, we did have a reference in the second paragraph where it said that a Robertsonian translocation was an example of a balanced rearrangement. So I'd jot that down in my outline. Furthermore, we should take a look at the results of this figure, the gametes. Keep in mind what a typical gamete looks like, because it may show up in the questions. Pulling it all together, we have a sample passage outline. Now, as you work on 7 through 10, pay close attention to the answer choices. Look for patterns, then ask yourself, how can I use that? You should make predictions whenever possible. While eliminating, look for those wrong answer pathologies and actually use the Kaplan labels. It will help keep your mental processes consistent. In number 7, we ought to notice something. There is some repetition in the answer choices. Number 1 is in all of the answers. That means it is correct. At this point, you can do one of 2 things. You can either forget about it because it's in all of the answer choices, or you can use it. We know that gamete 1 will lead to a viable normal phenotype. It's an example we can use to compare the others. What do we notice? It has a 14 and a 21, and this just might confirm your earlier prediction of what a normal gamete should look like. There it is. We may also notice that number 2 has a 14 and a 21. Granted, they happen to be translocated, but the necessary genes are there. At this point, we can use a little reasoning. The question asks us which gametes 1 through 6 will likely produce a viable normal phenotype. We'll notice that 5 and 6 are in none of our answer choices. So those are now examples of bad gametes. What do we notice about 5? It's got 2 copies of chromosome 14. And well, if that's bad, you can bet that 3 is also bad because it's got 2 copies of the 21st chromosome. So 5 is out and 3 is out, meaning we can eliminate B and C. Furthermore, we'll notice that 6 is an example of a bad gamete. Why? It lacks a chromosome 14, which also means that 4 is going to be bad because it lacks a 21st chromosome. So 4 is likely to lead to a zygote with a missing 21st chromosome. That means our correct answer must be A. In number 8, we have some subtle repetition in the answers. We have a mutation and a location. The STEM asks about partial trisomy, which we noted in our outline. The reference is in the third paragraph where it says partial trisomy refers to trisomy for only a portion of a chromosome. So which of these will lead to an extra copy of a part of a chromosome? A is out because that's a substitution. We need an addition. D is also out because it's a deletion. We need an addition. B refers to an unbalanced rearrangement, which you may recall from the passage. However, this is probably going to require a little more research, so let's check out C, which can be eliminated because inversions just change the orientation of a piece of chromosome and does not lead to an addition. So get rid of C, and boom, we have our correct answer. So without even needing to consider the entire answer choices, B must be the correct answer. On test day, the second half of each of these answers could be frightfully intimidating and beyond required knowledge. But that doesn't mean it's not fair. There was still sufficient information to determine the correct answer. In this case, all we needed was the type of mutation. You'll notice something different about number 9. We've been asked a question and we've been given a part of the answer, the part that is common in all of the answer choices. We now know that 35-year-old mothers are old and at a higher risk for trisomy 21 babies. But why? We're looking for a cause of trisomy. We actually noted this in our passage outline for paragraph 3, so going back and doing a little research, the passage indicates that most cases are from non-disjunction, so we're looking for that in our answer choices. We'd see it in answer choice C, and that is, of course, our correct answer. Number 10 has a classically structured set of answers, yes and no, with some logic. It's asking us about a Robertsonian translocation carrier. It provides some details and then asks about the zygote's risk for trisomy. It may help to picture the karyotype of our carrier. It should look something like this. We can see we have all of the essential DNA, even though we have the 21st and the 14th chromosome fused together. So are zygotes fertilized at greater than average risk for trisomy? Perhaps, but given the focus of our lesson, let's attack the answer choices. Anything in answer choice A raise a red flag? How about the word all? This is too extreme, and we could label this as a distortion. This is actually very similar to the second question we attacked today. This mutated chromosome is not going to be passed to all of the offspring. B is hard to argue against. If it were to lead to trisomy, then that would be a very logical reason why. C is tempting because the passage mentioned 5% of trisomy cases. It must be correct then. No. We're not going to fall for that trap. So what was the 5% from the passage? Approximately 5% of Down syndrome cases have the normal chromosome number with the third copy of chromosome 21 translocated. So translating that, we could say that 5% of Down syndrome cases result from a translocation. But that is not the same thing as having a translocation and then a 5% chance of developing trisomy. Think about it this way. If 5% of car crashes result from texting while driving, that is not the same thing as saying that if you're texting while driving, you have a 5% chance of crashing. Why is this one tempting? It is, of course, that detail from the passage. Under the time crunch of test day, this would be very appealing. D we might call out of scope because while the parent is phenotypically normal, we're asked about the genotype of the offspring. The parent is not genotypically normal. So with some savvy process of elimination, it's apparent that B must be the correct answer. So here we have a review of the wrong answer types. It's at this point when you want to start looking at your behavior to see if any of these are particularly tempting to you. Do you find yourself making a lot of miscalculations? If so, a math refresher might be in order. Perhaps you fall victim to the faulty use of detail. The key with these is that they don't answer the question. So it's probably in your best interest to paraphrase the question before moving on. Opposite answer choices may seem like they are the farthest from the correct answer. However, that's not necessarily so. Generally, they have language that is tempting, the content is relevant, and it's usually just a relationship that has been flipped. Sometimes it's a single word that'll throw those off as well. The out-of-scope answers can be avoided by making predictions. When you predict the relevant content, you're also predicting what is not relevant or out of the scope. Distortion questions take a true fact and then qualify it in a way that invalidates it. If we're going to avoid these, then we'll have to be extremely careful about any qualifiers, and we'll have to double check what we know and any facts from the passage. Now is the time to look for your own pathologies. When you begin to correct those, you'll get groups of questions right, and your score will improve.